Hello, I'm Merja Penttilä, a research professor in biotechnology at VTT and a part-time professor at Aalto University in synthetic biology. Biotechnology becomes increasingly important in the future in providing sustainable solutions that follow nature's principles. We can utilize biological mechanisms to synthesize products and can utilize the vast functionality we see in nature to make products that cannot be made with petrochemistry. Living cells and the enzymes they produce can be made into efficient cell factories. The novel methods of synthetic biology in particular give us the possibility to design these cell factories using computational tools and build the novel cells very rapidly. We design the cells using DNA as the code to do for us what we want them to do, to make novel materials such as bioplastics, chemicals, food ingredients, drugs, fuels, and so on. We are extremely happy that we have obtained substantial funding from the Jenny and Antti Vihuri Foundation to establish the Center for Young Synbio Scientists. This work is also supported by Aalto and VTT. The center includes a BioGarage where young researchers and those interested in going forward in setting a startup company can experiment and test feasibility of their ideas. We also want to communicate what synthetic biology is to a broader audience. And most importantly, we support young scientists in their PhD and postdoctoral work. In the following, you will hear the interesting research that our current students are doing.
Doctoral candidate Anna Ylinen is developing biopolymer production in yeast.
And I'm using synthetic biology methods to adjust the properties and making the production hopefully cheaper in the future. I'm producing especially PLA, which is polylactic acid, and then different copolymers with that, also polyhydroxybutyrate, BHB. Usually the lactic acid itself is produced with microorganisms and then it's purified and polymerized with chemical methods. But now we are producing both the lactic acid and the polymer already in the same cells in vivo.
Anna's research is based on the activity of various engineered wild-type PHA synthase enzymes. PHAs, or polyhydroxyl alkanoates, are natural polymers made by bacteria found both in soil and marine environments.
They are completely watertight, so it looks and feels like a normal plastic, but it's biodegradable and compostable.
Ylinen has transferred different bacterial and fungal genes to yeast in order to broaden the feedstock base.
I'm modifying the yeast cells that they would learn to use cellulose as a raw material instead of glucose. So we would want to use sugars derived from, for example, from cellulosic biomass instead. I've produced polyhydroxybutyrate, BHB, with cellulose, and I've compared two different enzymes inside the yeast cells. That one of them is using the cellulose a bit more efficiently and faster, but the other one would have more beneficial features regarding how the cell divides the energy inside it. So we are kind of trying to find the optimal way to use the cellulose for these purposes, but also for other applications.
Ultimately, this could even entail using industrial side streams as raw material.
The bacteria cells are efficient of what they do already at the moment, but yeast cells have a lot of potential for using industrial waste as a raw material, so for using new different carbon sources, since yeast tolerate low pH and harmful compounds very well, so maybe we can take directly some industrial side stream and we don't need to pre-treat it that much in the future.
Doctoral student Natalia Kakko is studying novel approaches for engineering cell metabolism and diverting cellular resources towards industrially feasible product levels.
The idea behind the improvement is that we want to turn off growth so that the resources that were wasted on the cells growing can actually be directed to the production pathways.
Kakko is developing an inducible synthetic protein degradation system for seizing growth and leaving resources available for product synthesis after sufficient biomass has accumulated. The research will demonstrate the use of this synthetic regulation system for heterologous muconic acid and glycolic acid production in yeast.
So the reason we're doing these specific industrial acids is because they're an important building block for a lot of more complex compounds, especially like the plastics that we use in like clothing or other textiles, different building materials, or further industrial compounds that are needed.
The novel approaches developed for improving yield and productivity are generalizable across products and extendable to other hosts.
Obviously, a lot of microbes are already being used industrially, but for the compounds we're focusing on, so like the industrial acids, the yields and productivities of the producers are quite low and they're not competitive yet. So what we're really trying to do is actually make them competitive with all the fossil resource dependent chemical synthesis processes.
Postdoctoral researcher Mariam Zabzevari is working on improving the design, build, test, learn cycle of synthetic biology with artificial intelligence.
Specifically, we use a machine learning technique known as reinforcement learning to design an iterative strain recommendation system.
Testing the performance of biological systems and learning from the experiments is still largely manual work and expensive, limiting industrial synthetic biology. The lack of automation in the DBTL cycle is a major bottleneck, and no comprehensive solutions currently exist.
Using organisms to produce chemicals on a large scale requires sophisticated computational approaches to control the behavior of a metabolism toward higher and faster productivity.
Mariam has developed a totally new computational approach to data analysis and creation of predictions.
It's a totally new algorithm that we have developed. So here the collaboration between biologists and computer scientists, because we need to know what will be the outcome of an organism if we regulate some certain enzyme levels in this specific way. And then based on the experimentation that they have done in the lab, we can recommend what should be the next experimentation that should be done in the laboratory. And basically we learn from those interaction with the cell environment that what should be the next strain that should be tried and could be promising in the sense of producing higher yields.
PhD student Feng Jianhui studies the mechanisms for self-assembly of biological materials inside the cells.
So we have known the liquid-liquid phase separation of the protein is an intermediate step for the assembly of the biomaterial, like the spider silk. But now I am more focusing on the biosynthetic material, which are produced from the microorganism, and I'm trying to understand how liquid-liquid phase separation happens and how liquid-liquid phase separations lead to the assembly of the protein molecule into a functional form inside of the cell.
Feng has developed a new method to study liquid-liquid phase separation using a model protein, the SUP35 protein.
The initial results show that the technique we are using, for example, the fluorescence microscopy and the super resolution analysis, and they can show us how the liquid-liquid phase separation of the SUP35 happened inside of the cell.
The aim of the research is to understand and control the molecular mechanisms that occur in the assembly of newly synthesized materials.
The optimal goal of this project is to help us design and then produce for example, some biosynthetic material with new and high performance properties. If we are able to see if the cell can form condensate inside of the cell, then we can only select the protein variant that can phase separate inside of the cell for the further purification. So that means you don't need to purify all of the protein variant you design. You only select the one that can phase separate inside of the cell. So that would save a lot of time and also increase some successful rate.
Doctoral student Robert Pylkkänen researches how a single enzyme can be used to produce cellulose without trees or even without cells.
We're making polysaccharides such as cellulose or beta-glucan, and these reactions can be carried in a test tube, so we can make cellulose in a test tube, which is quite exciting.
The tailor-made synthesis of these polymers is based on enzymes that break down cellulose in nature.
The enzyme was isolated from extremophilic bacteria, and what it did differently compared to other bacteria is that instead of using water to break down cellulose, it turned out to be a phosphorylytic enzyme. So it utilized phosphorylysis to break down cellulose. And then the difference there is that instead of breaking down the polymer to glucose units, we actually break it down to glucose 1-phosphate. So we retain in a way the energy of the glycosidic bond. And then also this enzyme can be used for the reverse reaction. So if we incubate it in the presence of glucose 1-phosphate, we can make cellulose.
The research has shown that the structure of these new materials, cellulose and beta-glucans, are unlike anything found in nature.
First you have the individual glucose chains that are making like this hexagonal chain, so it's always 6 glucose units per one turn around the helix. Those polymer chains, they come together and make larger structures, and also those structures are hexagonal in shape. But then we see that even those structures come together to make even bigger hexagonal structures, so they are making like multiple layers, and you have huge 10 micrometer in diameter hexagonal structures in the end, but it all starts in the nanoscale.
The application areas for these tailor-made celluloses and beta-glucans could range from controlled release drug delivery systems to photonics and additives in dyes and paints. Doctoral student Taru Koitto is studying microbial expansion-like proteins.
So my goal is to modify these proteins and sort of engineer them to be better in the modification of lignocellulosic materials or other biomaterials.
These proteins are naturally found in some microbes, but they're also present in plants themselves.
These are used when the plant grows, so that they can little bit expand the plant cell wall so that it is able to grow.
Expansion-like proteins are promising for this type of biomass modification, as they do not seem to have hydrolytic activity, which breaks the polysaccharide chains. Instead, these proteins affect the hydrogen bonds between the cellulose chains.
Cellulose itself has a nice structure. We don't want to break it. With these proteins, we could slightly change the structure of cellulose, but not without breaking it.
The expansion-like proteins are also studied in connection to Robert Pylkkänen's research on the cellulose synthesis. These proteins could contribute in the further development of Pylkkänen's material. Peshman Mohammadi is doing his postdoctoral research on the production of entirely new types of advanced biomaterials. The molecular design, assembly routes, and overall architecture of these novel materials are inspired by high-performance structural materials found in nature, such as the mantis shrimp club.
We have been studying mountain shrimp club. It's a material that has a really high mechanical strength, and high impact resistant. So in this work, our aim was to look into a possibility of mimicking some of the most essential features of the club to create a new type of nanocomposite material never described before.
Peshman has developed a robust biomaterial with high strength, stiffness, and fracture toughness by combining plant-based cellulose nanocrystals with proteins.
We actually designed a set of proteins that we have genetically engineered and recombinantly produced, which would not only reinforce the cellulose nanocrystal framework, but also regulate nucleation and the growth of hydroxyapatite mineral on the surface of the cellulose nanocrystal. So we have been able to make a multi-phase nanocomposite material from all these three critical components combined together.
These multi-phase nanocomposites are both lightweight and impact resistant, making them perfect, for example, for dental applications as well as other implants.
And this application for this could be expanded to, for instance, like in electronics, for instance, in photonics. But there's a lot of opportunity, I think, looking into interaction of different inorganic material and cellulose nanocrystal, and how CNC could be used to actually self-assemble, regulate the nucleation and the growth of minerals.